Hello and welcome to the last. Welcome to the last session of the international webinar on water health and parasites. In this last session, we have now the last worthy speaker, that's Dr. Shailja Singh. I request Shailja Singh to upload her presentation, and meanwhile, I'll take this opportunity to introduce her to the audience. Dr. Shailja Singh is presently working as Associate Professor in Special Center for Molecular Medicine and is Director, Advanced Instrumentation Center Facility at JNU Delhi. Shailja Singh is an example of highly successful, internationally acclaimed scientist in India. Her research contribution in the field of molecular parasitology is outstanding and significant, and majority of her work is published in high impact factor journals. Dr. Singh has received many awards for her research work. To name a few: Innovative Young Biotechnologist Award 2010, Senior Innovative Young Biotechnologist Award 2013, National Bioscientist Award 2018. Gandhian Young Technological Innovation Awards to her team in 2019. Dr. Shailja's extensive fundamental research on biology of human parasites, malaria and leishmania has shown a greater potential in translational research towards drug discovery and development. Recently, her work regarding malaria has shown a potential in improving the efficacy of an age-old anti-malarial drug, artesunate. A nanomedicine-based artesunate with surface-coated iron oxide nanoparticles has enhanced antimalarial potential in Plasmodium falciparum and its multi-drug resistant strain. This work has been published in eBiomedicine, the Lancet publication. To eliminate this deadly disease of malaria, blocking the growth and transmission both are equally important. Since Shailja's extensive research in finding the key protein which is involved in growth and gametogenesis can be applied towards the development evaluation tool to assist countries' malarial elimination effort. Her work in collaboration with Institute of Pasteur discovering the patatin-like phospholipase regulating perforin discharge and gametocide egress has a potential to be exploited to block transmission of malaria. This work has also been published in PNAS 2019. Recently, her group discovered a erythrocyte microRNA 1975P and found that it blocks growth of parasite with reduction in micronein secretion. PF apicotin acycloskeleton's protein expression was found to be reduced in microRNA-loaded erythrocyte. Overall, Shailja's basic research provides a platform to undertake translational research towards development of anti-malarial inhibitors and completely is in sync with malaria elimination by 2030. With this brief introduction of Dr. Shailja, I will now request Ms. Shailja to kindly initiate your presentation. Dr. Shailja, please.
Thank you very much for such a kind invitation as well as the introduction. My slides are visible? I'm in the first slide. Is it okay?
Yes, yes, ma'am, you are very much visible and audible also.
Okay, thank you. So,
thank you.
First of all, let me really thank to the organizers because such a short notice I've been included in such a great meeting, and it is a real good platform for me. Being a molecular parasitologist, I really like to share my research work as well as my whatever progress we have made towards the malaria as well as leishmania. And this is a real right platform for me to interact with the student and all my mentors as well as my peers which are there in the meeting. So to start with, I should share this, that why we really need to do some research in malaria, because already people are saying that we are getting less cases of malaria nowadays, and malaria is now coming very, very much down. So, but I really want to convince my audience that yes, still we need the malaria research, because you can see the right hand side, the malaria research, we are putting lot many funds, but then also in after 2018-19 we are not doing very well. There is a plateau in the decrease in the cases of malaria. So that itself describes us that yes, we need the research on malaria, because the parasites are becoming resistant to anti-malarial drugs which we are using. Along with that, even the insects which are the carrier for this malaria, like the vectors which transmit the parasite from one human to another human, that itself become resistant to insecticides. So in that case, we have a very less tool to eliminate this disease. So we really require lot many efforts again to do research on malaria. And for that, you can choose the field. You can go to the vector field, where you are trying to see, can you come up with a new strategy, how you can control the vector, control the mosquito. You can also do the research where you can come up with a new drug which half-life is good and those drugs are effective. They can also work in the resistant parasite. So on the parasite side also we need the research, but also at the vector side, where we want to eliminate maybe the mosquito which are carrier for the malaria parasite, there also we need the research. However, my research focus is mostly concentrated towards the parasite. So I'm trying to understand how this parasite biology is which respond to the drug, how this parasite try to become resistant towards the existing drug. Can I have the novel target present in the malaria parasite, and can I target those targets which are very much essential for the malaria parasite? Can I design some novel molecule? Can I inhibit those protein function, and for that can I develop a new drug? With that kind of questions as well as the objectives in mind, we are really working towards the blood stage of malaria. And in the blood stage specifically, we are trying to understand how these parasites are somehow growing in the human host. Just for my students who are in the audience, want to give a brief introduction about this malaria parasite life cycle. So you all know that malaria parasite, there are 5 human species present, which we know from the beginning, that 4 malaria, Plasmodium malariae, Plasmodium vivax, Plasmodium falciparum, and Plasmodium ovale. But the new species from monkey, earlier it was causing monkey malaria, but now it is also causing the human malaria, has come to the field. So in that kind, like we have 5 human species, and those 5 human species, 3 of them, they really cause lot of severity. Earlier we always had this in mind, that falciparum, they cause the all severity, like you get the brain fever, you get the all complications, organ failures when you are having the falciparum infection. But at the same time, now Plasmodium vivax also cause the severity, and those severity is very much similar to Plasmodium falciparum. So of course you would like to have the drug which are not only inhibiting the growth of Plasmodium falciparum, but you also like your drug which you have discovered that should also work into the vivax or the other species of malaria parasite. More importantly, not only this, but you will also like, for example, here in this slide, you can see the overall life cycle. How the life cycle goes when a mosquito, infective mosquito, which contains the malaria parasite, when it bites a human host, because it wants a protein meal, so the blood meal gives the female Anopheles the protein required for its egg laying capability. So for that, it goes and bites to the human host. And while this process, what happens, it injects some of the malaria parasite to that in the form of sporozoid to human. Then in human, when it goes to after biting, the parasite released to the human host, it goes to the liver cell. In the liver cell, it again proliferates, and then after proliferation, it grows and grows, and finally it becomes the liver cells release the form of parasite which we call the merozoites. Those merozoites come to the bloodstream of a human, and that bloodstream again, it sees the red blood cell, it talk to the red blood cell, meaning interact to the red blood cell. And finally, after interaction, it attaches to red blood cell, invades to the red blood cell, and finally proliferate and exit. So the cycle goes on. When do you get the all malaria symptoms? Only when from your liver malaria parasites are coming to your blood stage. That is the time you get fever, you get chills, you get all symptoms which are associated with malaria. That is why the blood stage malaria is important, because when you get sick, then only you will go to the doctor and you will talk to them for having the treatment. However, the liver stage time, there is no symptoms, but parasite is preparing itself to go further. But during the blood stage, some of the parasite, they also become male gametocyte, and then they became female gametocyte. And once mosquito again take the blood mean, these male and female gametocytes of malaria parasite taken by the mosquito, inside the mosquito, they again fuse and they form the zygote. Then they again go to the next stage, ookinete, oocyst, and finally form the sporozoite. And that sporozoite is there in the mosquito salivary gland. And again, during the bite of again the second bite of that mosquito to human, it has been released to the human. So for example, if you really want to have a solution, where will you block? Of course, you can tell me that yes, I can block at the liver stage, but again, at the liver stage, you don't have the symptoms. How will you know the parasite is there in your liver? So that means when the real symptoms are appearing, that is the target point. Basically, the blood stage are the target point where you are going to interact. But however, for the vaccine, the vaccines, they work in terms of the prior to infection. So in that case, you can make a antibody in the liver stage, and in the liver stage, you can have the inhibition in the liver stage growth. But for the chemotherapy, it is always the case, you will look for the blood stage. But at the same time, you would also like to kill the parasite in the mosquito stage. Why not? Because maybe you want that, you don't want to kill the mosquito, but you want to kill the parasite inside the mosquito, because the ecosystem we don't want to disturb. So can you have this strategy? Can you have any innovation where you are really killing the parasite inside the mosquito, but not the mosquito? So in that case, now the genetic approaches have come. We have generated some of the microflora, that is like some bacteria. These bacteria are present in the mosquito, and whenever they see the parasite in the mosquito, they kill the parasite. So in that case, you save the mosquito, that is the blood feeding arthropods, but you are killing the parasite. But that is, again, keep this apart. You would like to have some chemotherapeutic agent where you would like to kill the parasite in the mosquito stage also. So looking at all this aspect, what do you want to do? At that point, you would like to have one protein which is there, this protein of malaria parasite, which is important, and also there in the liver stage, they are there in the blood stage, they are there in the sexual stage of malaria parasite, as well as if they are there in the mosquito stage. So you can have one protein which you will target to kill all the stages. So it is too ideal to achieve, but we researcher really, we would like to have this. So in this case, this is one protein called perforin like protein, which we have identified, we have discovered in malaria. So this malaria parasite expresses this perforin like protein in the liver stage also, in the blood stage also, in the gametocyte stage also, and also in the oocyst stages. So that means it becomes one target, but targeting the all stages of malaria parasite. So what is this perforin like protein? This perforin like protein, as name suggests, it forms the pores. So why it forms the pores? Whenever like in the left hand side, you can see these perforin like proteins are monomer, but when they see the other monomer, they form the oligomers, and those oligomers, it whenever it sees the membrane, it inserts to the membrane. And once it inserts to the membrane, it again forms a hole in the membrane, and that holes are deleterious for any cells. So in that case, this is some kind of a strategy which is been developed by malaria parasite for its own survival process. But you can ask the question, why the malaria parasite has this perforin like protein? But as I explained to you, that any stages malaria parasite goes to the host, it uses all the host material to grow, and finally it has to come out from the host to infect the new host cell. So it should have some arsenal, it should have some tools where it should help malaria parasite to go in, and then some of the strategy it should have which will help coming out from the red blood cell. For example, you can take host as a room. So malaria parasite is entering to the room with some of the tools, and then it is using whatever facility in the room, and finally when it is exhausted, the parasite is grown enough, it will come out by again having some strategy or the tool. So the tool itself parasite expresses is the perforin like protein. And then how many? There are 5 perforin like protein, PLP 1, 2, 3, 4, 5. You can again ask me a question, why there is 5? Because these are all members of the same family, but they are present in different stages. Maybe some proteins are present, some members of these proteins are present in the blood stage, so helping malaria parasite to maintain the blood stage. Some proteins, some members are present in the sexual stage, like a gametocyte. So those protein members are helping parasite to for the fusion, for the exit from the gametocyte stage, and then maybe they are also there in the liver stage, so they are helping parasite to come out from the liver stage. So that's what this protein becomes so important, because it is there in the parasite. There are 5 members, and all 5 members are present in the different stages. So first thing we wanted to do is that let me characterize this protein. So one of my PhD student, and later on she has got the Inspire Faculty Award also, she started thinking in that direction, how one can characterize this perforin like protein. So for that we cloned this gene from the malaria parasite, we made the protein, and again it was a bit difficult to make the protein because protein itself is toxic. Whenever it sees the membrane, it might damage the membrane. So we used the E. coli system to express this protein, and finally we were very, very lucky to make these 2 perforin like protein 1 and 2 in our E. coli system. So then you can ask me the question, like what was the different strategy which we used to make this protein. So the strategy was to, because this protein, when it is in very diluted form, it doesn't damage the cell membrane. So while expressing, what we did, we were exchanging the media, we were collecting the media in between. So we were very much sure that yes, the protein we are harvesting, it is not damaging to the expressing cell. By doing this, we could make the protein. After making the protein, the biggest challenge was, show me that this is a perforin like protein. How will we show? Because if we are claiming this protein is a perforin like protein, we have to show that it forms a pore. So how you will visualize the pore? There are many methods. You can go for transmission electron microscopy, you can do the atomic force microscopy, because that gives you the real structure of pores. So here you can see in this slide that we took the protein, we took the red blood cell, we mixed them together, and finally we visualize under the atomic force microscopy, and very nicely you can see the pores on the membrane. That was our eureka moment, because we could see that whatever protein we have made coming from malaria parasite, now it is really seeing the membrane, and what we expected that it will form the pores, it was forming the pores. But we also measured how big the pore is. Then you can see the height and the diameter which we measured, and then because it forms the pore, ultimately it lyses the red blood cell, right? So to see the lysis of red blood cell, the process is very simple. How any human red blood cell are lysed? whenever you take the red blood cell, you put the water, because of the hypotonic solution, red blood cell burst. So that the release of hemoglobin becomes the 100% lysis thing. But you can also incubate your red blood cell with this perforin like protein, and because of the lysis of red blood cell, there will be hemoglobin release, and that hemoglobin release can be measured by a spectrophotometer. The spectrophotometer reading is also very simple. Hemoglobin gives the absorbance at 405. So a spectrophotometer will tell you how much hemoglobin is been released, and you will have the parameter to see how much lysis is there. And then that lysis will tell you the perforins are working or not working. Here I will show with all this relevance, once we characterize our perforin-like protein, the next thing to make some context that really if this perforin-like protein is working for malaria parasite biology, is it helping malaria to exit out of the red blood cell? So in this movie, we knew that perforin-like protein, they are calcium sensitive because their oligomerization is calcium sensitive, their insertion to the membrane is calcium sensitive. So we first wanted to check if at all there is a role of intracellular calcium for the exit of malaria parasite. And so I'm going to show you this movie, and you will really appreciate, because this took me almost 6 months to make this movie, where I have taken whatever in a human body, you see the process that parasite is inside the red blood cell, and that red blood cell, when the parasite matures and enough, and they decide to come out, how it break open the red blood cell to come out, and at that time point how the calcium dynamics is changing. So how you can measure the calcium dynamics? There are some indicators which bind to the calcium and they fluoresce. So here is the two-dimensional live cell video microscopy, where you can see the parasites are coming out, breaking open the red blood cell, and their calcium is also high. So you can see here in this movie, yeah, there was a burst and there is a calcium increase. With that, I asked the question, if there is a burst of red blood cell to for the parasite to exit, and there was a calcium increase, if at all, I chelate the calcium, I will not allow the calcium to go up, whether my perforin like proteins will be active or not active. And if they will not be active, are the parasite are locked up inside the red blood cell, and if they are locked up, not being released, what will happen? They will be cleared in human body, so they will not be able to proliferate. So to show that, what we have done, we took the parasitized red blood cell, we chelated the red blood cell with the calcium chelator, and then finally look for the perforin release. So the green dots in the image shows the perforin, and you can very well see that the red blood cell membrane has the green dots. What it represents? It represents the perforin-like protein. So in a normal condition, there will be calcium, there will be release of perforin, it will go to the membrane, it will make the pores, and the parasite will happily exit from the red blood cell. But if you have chelated the calcium, there will be no release of perforin, there will be no pores on the host red blood cell, parasite will be locked. And that's what the BAPTA AM, the down panel shows. There is no exit of merozoites, that is malaria parasite. Again, while doing this, I was very much interested to see, okay, what happens to the cells where the parasite is there, but what happens to the cells where parasite is trying to invade. So in this movie, you will see that the green thing is the exit of malaria parasite, but nearby red blood cell, in that you will see a parasite is attacking to that red blood cell, and that target red blood cell calcium is also going high. And this is really interesting again. So there is a burst, and then one merozoite tried to invade, it invaded, and then the calcium went high, and then again the calcium goes down. And then you can see the red blood cell became very echinocytosed, meaning it's trying to accommodate the parasite. And finally, what happened? It comes to the normal, a relaxed position, because the parasite is already entered. And this is also happening in the body, but we could see it under the microscope, and thanks to the technical advancement we have. So just for your understanding, I have shown the screenshot of this. But at the same time, I also want to propose the model that where comes the role of perforin, because here I am proposing that maybe this perforin like proteins are being secreted by malaria parasite and goes to the target cell, and because of the target cell entry, the calcium of the target cell goes high and helps parasite to accumulate. Can I prove it? Yes, I can, because if I will chelate now the target host cell calcium, maybe I will not see the invasion, and that was the case when we did the experiment. And finally, what we proved, that the echinocytosis process of the target red blood cell is being mediated by perforin-like protein release, and finally it helps in the malaria parasite invasion. So the last time I showed was the egress, and this time is the invasion process. But we again did the very like a very extensive study, because we wanted to show what happens, like really this perforin like protein, whether it is being secreted, it is targeting to host red blood cell, is it inserted to host red blood cell. So you can see the images where the red uh, color shows the red blood cell membrane, green color shows the perforin like protein, and you can see the x and y angle, that yellow color, that means the protein is inserted to the membrane. So what we see in the picture was really happening, and this was the case during the invasion process. Now you can again ask me the question, okay, if you are showing me that during invasion this is happening, you have the protein, now you show me that it is really happening when you are taking the red blood cell. So my own red blood cell I took out, and here is the movie again, and we these red blood cells were incubated with the perforin like protein, and we also labeled this red blood cell with the calcium sensitive dye fluo 4am. So when I will run the movie, you will see now you can't see any green, right? Everything is like a red blood cell, very nice concave shaped red blood cell. But as soon as, and we have added the perforin into it, but when we are seeing under the microscope in a live cell mode, you can see how this is going to go green. Now you can see with time these red blood cells are getting green, and those after getting green, meaning calcium increase, they what happened, they echinocytosed. Like you can see now they are crenated, they are not very beautiful, meaning some echinocytosis process, and swollen also. And then this is the process we wanted. This is happening because we have added into the red blood cell our perforin like protein which we made. But this is also happening in the regular physiological term, where we just use the parasite. That parasite will secrete, and that secretion will target the red blood cell. So just to show you this happens, we have done this experiments. So with this movie, it is again a snapshot. You can see earlier in 0 minute we could not see anything, but then it became green, and then the cells were echinocytosed. But at the same time, you will ask me, show me the perforins are there. So I could stain with the perforin like protein antibody, and all were there very well decorating the red blood cell membrane. And but these red blood cells, what is happening to them? Because they have now got the small pores, which I showed to you by atomic force microscopy, the previous slide. They also become annexin positive, because now they are not healthy enough, and that's what we have done the annexin stain. Now you can again relate this data which we are generating in vitro in a real term. You know that whenever we get the malaria infection, what happens? We get anemic. Meaning what? The anemia, because the red blood cells are disrupted. In our blood, these red blood cells, why parasite is infecting one red blood cell, but why my other red blood cells are now lysed? Because of this perforin release. And that's what we could show. The anemia, the severity of malaria, one severity of malaria is very severe malaria, and that we could relate with the discharge perforin. Now not only the human red blood cell, but also like we have our blood capillaries, and the blood capillaries has the endothelial cells. So if the parasites are secreting this perforin like protein, what happens to those endothelial cells? That was the question we asked. So we grew the endothelial cells and we added the perforin into it. And you will be very surprised to see this movie where the cells, the endothelial cell of human, like they become very, very apoptotic, necrotic, and first step is the blebbing. So you can see this cells now, and we have added the perforin again. So what is happening? You'll be really enjoying this movie. You can see how the membrane dynamics changing, and then there is a blebbing process, and this blebbing is being created by the addition of perforin to the endothelial cell, and finally these cells, they become very, very fragile, and finally they die through the necroptosis mostly. But again, at the same time, we also wanted to I'm showing you this blebbing, the snapshot of the blebbing, because apart from the live cell, we also take the snapshot through the confocal microscope, and you can see the earlier the cells was very nice, but later on the blebs are very much there. But when it is happening, is it calcium enhanced? And you can see again a movie in this cell that there is an increase in calcium, there is a decrease in the survival, and that survival is being seen by the propadium iodide positivity, because we know if the membrane is compromised, propadium iodide is going to enter. So we all know that dead cells are being labeled through what? Through the propadium iodide. So in this case, what we have done, we have taken the cells, we incubated with perforin like protein, we also added the calcium sensitive dye, we also added the propadium iodide, that is a cell death indicator, and you can see how the cells are going to die when we have added the perforin. So here in this movie, what is happening? Uh, Now they are green positive. Now they are going to become the red positive, and you can see even the blebbings are quite prominent. That means what? That means the cells are really making all endothelial cells die. And that's what it is being linked with the severity. Now the question is, you have to prove it through the other molecular tools. You can label it for the propadium iodide, you can label it for the JC1, because we know that any endothelial cells, when the mitochondrial membrane potential is lost, you might get some indication that the cells are going apoptosis or the necrosis or anything. So that way you can see. So we labeled, so we incubated the cells with the perforin-like protein, and then we look for the annexin PI staining. Live cell I have already shown. This is the snapshot of those, and also the JC1 staining. So you can see what is JC1 staining. This is again a dye which indicates you the health of the mitochondria. So when it shows the green, meaning in that case the red, when the dye enters to the, if the membrane potential of the mitochondria is intact, JC1 dye enters to the mitochondria, and that mitochondria, because the health is okay, membrane potential is okay, it stains red. But if membrane potential is lost, the dye comes to the cytosol, and then it becomes a monomer, and that dye shows the green. So you can see in a healthy cell, in a control cell, there is some good ratio of red and green, but in a perforin-like protein treated cells, that is down panel, you can see the red is low, but green is very prominent. That means the membrane potential is lost. We also calculated that, and then finally we come to know that yes, this perforin-like protein, when we incubate with the endothelial cell, it is killing the endothelial cell through the apoptotic process. But we cannot conclude just with in the form of annexin PI staining that it is the apoptotic cell. You know, there are multiple types of cell death processes: apoptotic, necrotic, necroapoptotic, or you can say anoikis. It's a new term. I think you all know that there is a one cell death process being known as anoikis. So there could be any pathway being triggered by this. So we also wanted to see whether it's a necrotic death or the apoptotic death. So one of the marker for necrosis is what? HMGB1 staining. So when the HMGB1 is nucleus, that means there is no necrosis going on. When it goes outside the nucleus and go to the cytosol, it indicates that there is a necrosis. So you can see in the control, the blue color, which is a DNA or the nucleus of the cell, and the green color, which is HMG1, HMGB1, they are co-localized. So you can see there is no necroptosis. Why in the control you will see any necrosis, but in the RPLP2 treated, meaning perforin treated cells, HMGB1 is been released from the nucleus to the cytosol. You can see the blue dots separately and the red are spreaded. You can do this analysis by any confocal microscopy, and you can make the linear graph. So where is the color, the green color and the red color, and how you can do. So it is very simple. If anybody wants to clarify, I can answer you that also. But the next thing is, whatever this biology is, ultimately you have to come with a solution that yes, this perforin like protein makes pores, helps parasite to proliferate, and also very dangerous for the human health, because whatever severity being caused, endothelial problem or the red blood cell severe anemia problem, it is because of this perforin. You should have some solution of this. So to have the solution, we also wanted to design some small molecule which should block this perforin like protein. So how you can block this perforin like protein? Either we should design an inhibitor in such a way that it should be kind of a patch to the pore. So some kind of a, if you see in any house there is a hole, you will try to cement it to have the pore closed. Similar way, like first we wanted to see through in silico, can we design some small molecule which are fitting to the pore region of the perforin like protein, and for that we modeled this PLP. And as I told you that this PLP, they form the oligomer, so we also modeled these monomers to form the oligomer. And then we talked to our chemistry collaborators. We showed the structure and we said, can you please design some molecule which will bind to these perforin and should block the activity of the perforin. And you can see here, they designed some molecule which was some kind of a interface between the two molecules. So one perforin and the second perforin monomer, when they bind, and that's the way they form the oligomer, this molecule never allowed to form the oligomer. So if they will not be in the oligomer form, they will not enter to the membrane, and finally the membrane will be saved from the lysis, as well as the parasite will be locked inside the red blood cell, and the parasite will also not be able to enter into the red blood cell. So all processes. So first thing, if I am designing a molecule, I need to show whether my molecule which I have designed, it binds to the monomer or not. What are the process to show? Small molecule or any compound binding to protein, there are multiple methods like ITC, isothermal calorimetric. There are process of SPR, surface plasmon resonance method. So those methods, these are biophysical techniques which tells you, yes, there is a binding of your molecule with the protein. So we use this SPR method, and then we made this perforin-like protein. We incubated the two. We put this perforin on the chip, and then we flow the inhibitor, this C01 and C02, the inhibitor, we named it like that. And then we could show there is a binding constant being appeared, and that you can see in the graph, when as we increase the concentration, there is more and more binding, and then it becomes saturation. So with this, we were very happy that at least we have some molecule which can bind to perforin. But just binding is not inhibiting the activity. So to confirm that it binds, and then it also blocks the activity. So how you can see the blocking activity? I already told you that perforin-like protein, when you incubate your red blood cell, there is a hemoglobin release. So we did those experiments in the presence of inhibitor, and you will really appreciate the inhibitors. They could block the lysis of the red blood cell. And we also showed, then you can say, okay, is it lysis or the pore? So again, I could show to you that how you can see the pore, either by by examining under the atomic force microscopy, you can see there is no pore. Or you can use some indicator of a specific size, and you can ask the question, if there will be pore, inhibitor will enter and label the membrane. If there will be no pore, there will be no entry. So you can see in the control, no entry, there is no red color. But in rpmd, the molecules which perforin, when we incubate it, the membrane becomes red. Why it becomes red? Because we have put the dye, and dye goes inside and then it labels the membrane. But if we have added the inhibitor, again there is no red, meaning the pores are not been formed. Why the pores are not been formed? You can ask me again, I have answer, because there is no oligomerization. And because there is no oligomerization, there will be no insertion of this perforin to the membrane, and there will be no pore formation. So all this scientific proof we have for our data. And then we also looked under the microscope. Let me check whether the pores are being formed or not formed. So you can see in control red blood cell, there will be no pore. But when we incubate the red blood cell or the erythrocyte with rpmd, the recombinant perforin-like protein, you can see the pores are all over the membrane. But if we incubate the protein with the inhibitor, again there is no pore. So that itself is telling me that yes, my inhibitors are protecting the red blood cell membrane against this parasite protein, perforin-like protein. So, but finally, I need to show whether these molecules are anti-malarial or not, because ultimately malaria severity has been caused by perforin. Malaria growth and invasion egress is caused by this perforin-like protein. And you can see, yes, these two compounds are really good anti-malarial. They are blocking the growth of malaria parasite, right? So you can see in the percent inhibition with these two compounds are really good. They are in some micromolar range. They are blocking the invasion and egress, and then formally the invasion per egress. Now, because they are blocking the blood stage growth, my next question is, are they blocking the liver stage growth? So for that, what we did, we used the mosquito sporozoite. We dissected the mosquito. We took out the parasite. We took the liver cells. We put the parasite in the presence and absence of this inhibitor. And you will be really happy to see that we could block the liver stage growth. That is my left-hand side panel. And then it also blocked the gametocyte stage, because the gametocyte also, they need to come out to fuse to make the form zygote. So there in the right hand side panel, you can see that my compounds, the C01 and C02, are blocking the gametocyte egress also. So with this, what am I proving? I'm proving that yes, I have designed some good two molecules. They are blocking the growth of malaria parasite in the liver stage, in the blood stage, and also the sexual stage, that is mosquito stage. With that, I also wanted to see whether my compounds are blocking the lysis of red blood cell or not. And you can see when we put the compound, our compound with the red blood cell, and then we add the perforin, our red blood cell are very much fine. They don't echinocytose, they don't lysed. So again, a good news that maybe the severity by anemia can be blocked by these two inhibitors. Then also there is no annexin positivity, because red blood cells are really protected with the compound. So the compounds are making the red cell. And also at the level of eruptosis. So like in normal cell, like endothelial cell, we say it's the apoptosis or necrosis. But red blood cell lysis is known as a term eruptosis. So that is itself is being saved. Our red blood cells are not going through eruptosis process. They are saved. That's what I want to show in this slide. And finally, I would like to acknowledge all my members, my group members, because it is their hard work, my students with all smiling faces. It is their hard work where we could achieve these many good research as well as the good science. And they are really inspired and they are really motivated to do good science. And I'm very lucky and fortunate to have them. I also want to acknowledge the government Department of Biotechnology, Department of Science and Technology, the other funding agency like National Institute of Health USA for funding me to do this kind of a research. Finally, I would also like to thank the organizers again to give me the opportunity to present my work. Thank you.
Thank you very much, Dr. Shalja Singh, for this thoughtful presentation and path-breaking research on the biology of human parasites. Through your lecture, you highlighted the role of parasitic factor PLPs in dysfunctioning various barriers of cells, including endothelial cells. So, understanding the root cause of the severity of parasitic pathogenicity of Plasmodium falciparum, you can not only contribute academically but also serve humanity by proposing drugs or therapies for total eradication of malaria. Thank you very much for this valuable and novel research idea. Thank you very much, ma'am. Ma'am, with your permission, we have received certain questions.
Yes, please. Yes, please.
uh, Thank you, ma'am.
Can you read the question for me?
Yes, ma'am, yes. Ma'am, first question is, can natural selection be considered as one of the major risk factor to develop drug resistance in malarial parasite? If so, how?
Yes, of course, because you know that natural selection is real, the basic for
Anonymous.
any resistance. So this is the important point, and this is well established and well accepted fact that this is the root cause for the resistance. But if at all we would like to even fight against this resistance, what should be our strategy? The strategy should be that we should be targeting the host, not to the parasite. So then the parasite will not be having any control. So in that case, that's what now the drug strategy has been changing from the parasite targeting to the host targeting. And we are also working on that, because in that case, you don't get the resistance soon, because the ultimately the red blood cell is the host, and if I am targeting the red blood cell, how come the parasite will be having any access to human red blood cell to change something for its protection? Yeah.
Okay, there's another viewer, Rinchen Angmo from Ladakh. He wanted to know whether malaria outcome or virus depends on climatic conditions of the place.
So malaria is not been caused by the virus; it is been caused by the malaria parasite. So it's the parasitic disease, and basically climate is quite important because if the vector for malaria is the Anopheles mosquito, and these Anopheles mosquito, they are not there; they are very much dependent on the climate and the weather. So earlier it was like. We were thinking that these mosquitoes, they are day biting mosquito, and sorry, they are night biting mosquitoes, so we were saving ourselves by having the bed nets at the night. But now these mosquito have changed their behavior. Now they have become the day biting mosquito, so I cannot go out with the mosquito bed net. So of course the climates and everything is these are having a major role for any disease, as well as the malaria. But at the same time, earlier we knew that at the very high temperature there will be no mosquito spores. But then later on we found that even at the higher temperature they can resist, and there could be some mosquito. The similar way, maybe at the lower cold condition we might find some mosquito. So ultimately, because of the change in the behavior of the vector meaning mosquito, yes, we are not able to eliminate this disease. But certainly, the climate has a role to play.
Thank
Yes,
you. Another question from Professor Mohammad Niamat Ali.
yes,
He wants to know molecular test you did for necroptosis,
yes,
and
yeah.
how your contribution will be helpful for next generation anti-malarial drug.
Yes, so thank you very much. So for necroptosis molecular marker, as I said, basically you have to prove that some markers from apoptosis and some markers for uh, necrosis. If you are finding in your cell both, for example, HMGB release, some of the caspases, if they are activated, then HMGB release. So if you get the mixture of both markers, apoptosis marker as well as necrosis marker, you can conclude that there is a necroptosis going on. So the necrosis plus apoptosis is going on, and there are some good papers in Cell Press have come only on this necroptosis processes. And the second question was on elimination. What was the second question? Sorry.
That was how your contribution will be helpful for next generation anti-malarial drug.
Yes. So just to, I'm really fortunate enough to be as an expert in medicines for malaria venture. So my contribution not only in the biological science but also in terms of the novel ideas and the innovation where it can be put towards a expert all across the world. And this MMV host around 40 to 50 countries participating, and I'm the expert member in that. So my contribution is to have the good biological information, and with that good biological information can anybody design the good drug. And what do you mean by good drug? The good drug meaning maybe their property, their half life, their other properties, and their efficacy, as well as their resistivity. All those parameters are very much wishful way, meaning what we want they should not create any resistance. We want there should be more time to the body. We want a single dose. We don't want to give our children who are suffering from malaria multiple dose. So all those are wish list, and my contribution, I think, has been recognized by the world, and that's what I am now the part of the medicines for malaria venture. Yeah, thank you.
Of course, ma'am. There is another question from Dr. Samiullah Bhatt. How
Yes.
should genotypic, another question from Dr. Samiullah Bhatt. How should genotypic homology of recurrent Plasmodium vivax interpreted in epidemiological studies? Your comments, please.
Yes. Basically, on epidemiology, molecular epidemiology work, like what I know from the field that the Vivex, basically we have designs very specific probes or SNP specific to the Vivex and the falciparum. But we always in the epidemiology we get the co-infection. So it's very very tough to have only the falciparum infection or the Vivex. In India also we have both, like you get the falciparum and then also you get the Vivex. Maybe the falciparum severity is more, so you are treating the patient with the falciparum treatment, but at the same time if you have the molecular diagnostic proper and the diversity is you have kept in mind, you will also see the Vivex infection, and this is the problem. That is why we are suffering more towards the elimination process. Yes. And you know, in the Vivex, we know that it is there in the liver stage for months together, and then suddenly relapse happens. So of course we cannot diagnose when it is there in the liver stage. So if you can design some molecular diagnostic when the parasite is there in the liver and you can really identify it, that would be the real good challenge for molecular epidemiologist. Yes.
Okay. Another question is from Zubair Hassan. What is the significance of the association between malaria and sickle cell anemia?
Okay, so it's quite well obvious, like we know that sickle cell anemia, because of their shape as well as their membrane rigidity, they are resistant to malaria. But you know, this is now being challenged. Now we know that all cyclic patients are not malaria resistant. Rather, the severity of malaria is more to the cyclic patient. So like things are now changing. The like I was talking to the New York blood bank research head, Dr. Mohan Das Narla. He has spent a lot of time on sickle cell anemia and malaria. So earlier we used to know, okay, if the patient is cyclic or sickle cell anemia is there, maybe they will not get the infection to malaria. But now the things have changed. Maybe the patient with the low parasitemia will show the more severity, because they are already fragile. Those red blood cells are already fragile. Maybe the parasite will enter, and little bit of infection can make more and more lysis of the red blood cell. More and more severity might happen. Yes.
Okay, another question is from Zahid Wani.
Yes.
He wanted to ask, despite abundant evidence that PPLPs are crucial for life cycle progression of malarial parasites, why development of chemotherapeutic interventions against them have not been explored?
Which one? Which target he has asked? I'm sorry. PP.
PPLPs. PPLPs.
Okay, okay. So yes, see, the problem is this perforin-like protein, which we have shown, it is present in the multi-stage malaria, and by targeting it, we can treat the malaria parasite. But you know, as we develop, similarly, parasite also develop. So it's so clever that it doesn't depend only on the one kind of a machinery, one kind of a pathway. So as soon as we come up with one solution, it has the other method to go. So that is why the intervention strategy, yes, it is a good contribution from my lab, but maybe the next thing will happen that the parasite itself is going to change a lot. So maybe it will change some mutation in the perforin-like protein. Maybe its expression, it is going to be changed. Maybe in the field something will happen, and maybe this intervention will not work that efficiently. So yes, this is a challenge. One should keep that also in mind.
Okay, another question from Ali Murtaza. He wanted to ask, is malarial medicine hydroxychloroquine
Yes.
effective against the COVID-19 virus?
Yes, so we have been reading in news that hydroxychloroquine is basically a good drug to be proven by for against the COVID-19, and I think up to certain extent it is true because some trials been made, and this hydroxychloroquine, because of its impact on the host. So the when the virus is infecting to the host cell, and there the target is like all the autophagic process which is being used by the viruses are being targeted by this hydroxychloroquine, and finally you see the reduction in the proliferation of the virus. Yes.
There's a last question from Muzaffar Mahideen. What
Yes.
steps should be taken and are useful on domestic basis to prevent ourselves from malaria?
Yes, so the first and foremost thing I should be, of course, the mosquito control. That is our domestic thing for sure. That we should not allow the mosquito breeding nearby area, because these mosquitoes are the vector. But at the level of other anti-malarial herbs and other natural compounds, I think in India we are fortunate to have many natural compounds around us, and that's what it happens. Like I'm collaborating with Nigerian scientists, and there you won't believe that parasite load is too much. Every child has this malaria infection, adults as well as the old ages people also have malaria. And for that, generally like every month they have to take the all this chemotherapy, but at the same time they also take some Nigerian plant extracts to control the malaria. So we should learn from them, and we should also do for India that if we know that some of the natural products are good enough, we should take those measures as well. Yeah.
Thank you very much, Dr. Shalja, for your illuminating talk and novel research idea for getting medicine for this deadly disease, malaria. Thank you very much for, ma'am, for your this elusive talk.
Thank you.
Before I wrap up, I, on behalf of the organizing committee, are sincerely grateful to all the worthy speakers who spare their valuable time despite their busy schedules and share their valuable experiences and novel ideas and research. We really got enlightened. I'm also deeply grateful to all our audience listeners for their patience and connectivity throughout the webinar. Thank you once again from my side, but you're requested to stay tuned for valedictory function, which will be hosted and moderated by Dr. Muhammad Muslim. Assalamu alaikum.
Yeah, assalamu alaikum. Yeah. Yeah, just stay tuned. We're just waiting for the rest of the panelists so that we can start the validity session.
Just wanted to check my screen share is off, right? This is Shalja. I just
Yes, ma'am. It's off.
thank you very much.
Okay, Bismillahir Rahmanir Rahim. Assalamu alaikum wa rahmatullahi wa barakatuhu. I welcome you all to this validity session of the international webinar on water health and parasites. I'm Muhammad Muslim, Assistant Professor, Department of Environmental Sciences, and I'll be moderating this validity session for you. To begin with, I request Professor Fayaz Ahmad, Director Webinar and Head Department of Environmental Sciences, University of Kashmir, to present his concluding remarks. Professor Fayaz, please.
बिस्मिल्लाहिर्रहमानिर्रहीम। अपने भी खफा मुझसे हैं बेगाने भी नाखुश, अपने भी खफा मुझसे हैं बेगाने भी नाखुश, मैं ज़ाहिरी हिलाहिल को कभी कह न सका कन। अस्सलाम वालेकुम and very good evening to one and all.
इसको दे के आ जाना।
A hearty welcome to this valedictory function of the 3-day international webinar on water, health and parasites. This was an integrated webinar wherein we tried to have speakers and panelists from across the world, across the globe, to speak on varied topics, as is clear from the 6 technical sessions, because in all we had 8 sessions, inaugural valedictory, and then 6 technical sessions. We had 14 speakers, 9 of international from different countries of the world, including Professor Zhang Fan from China, Professor Basant Maheshwari from Australia, Dr. Amit Kumar from China, Professor Marco Cantonati from Italy, Dr. Binayara Shivakoti from Japan, Dr. Samindra Sherchan from USA, Dr. Larry Stevens from USA, Dr. Sara Naureen from Bangladesh, and then Dr. Mohideen Munawar from Canada. We had 5 speakers of international repute from the country, including Professor Sandeep K. Malhotra, Professor Umi Shamim, both of them vice presidents of the Indian Society for Parasitology. We had Professor Kuldeep K. Sharma, Professor C.J. Haware, and just now we had Dr. Shalja Singh, who all of whom were somehow novel in their ideas and in their works which they presented to the audience. And all the 3 days, we had participants not less than 200 at any point of time. Although 510 participants were registered, but as we see in the seminars and in the webinars, that majority of the people don't join. But in this very particular webinar, a unique feature was that majority of the people were connected and majority of the people were enthusiastic in learning more and more from the panelists. And the number of questions was so large that at some point of time, I had to intervene and request the moderators to please cut short the number of questions. That was the level of enthusiasm among the participants. In the inaugural session, which we conducted on the 22nd of this month, that is on Monday, the Honorable Vice Chancellor, University of Kashmir, Professor Talat Ahmed, inaugurated the webinar and presided over the function. And then we had the president of the society, who was the chief guest at the occasion, Professor Subir Kaur. Then we had our Dean Research, Professor Shakil Ramshu. Then we had Dean of our school, Professor Shamim, in the inaugural function. And then today, in this valedictory function, we have our revered Dean Academic Affairs, Professor Shabir Ahmed Bhatt Saab, with us. Then we have with us the General Secretary of the association, that is Indian Society for Parasitology, Dr. J.K. Saxena Saab. And then we have the Controller of the Examinations and Dean School of Biological Sciences, Professor Irshad Nauchu, with us for this valedictory function. We have a very elaborated report which we have prepared, but probably it won't be feasible to repeat and to say all the points which were raised and which were deliberated during these 3 days. But one thing which I would emphasize over here is that all the speakers highlighted such areas which opened new areas of research and which obviously made the young researchers very curious about having new thoughts and new areas wherein they would now start working on. And the unique feature among the learned speakers which they presented during this webinar was that they have the data for a lengthy period of time, long period of time, that actually makes any type of research work very effective, that we compare the data for the past so many decades, that somehow gives us a clue that what was the status earlier and what is the current status. Then the ideas which were floated for the youngsters, and then the certain learned speakers put forth the techniques and the pattern which should be imbibed and which should be followed by the researchers in say putting up the projects and in writing the research themes and in somehow going to the newer areas of research, so that they would independently carry certain research and they would independently be in a position to achieve funding. Since we had selected water and parasites as two main themes for this very particular webinar, and all of us know the importance of water, and all of us know that with each passing year, we obviously are facing and will be facing the shortage of water. So how important it is to work on this very particular area, the water conservation, for the sustenance of the human race. And then on the other side, we have the parasites, and as has been spelled out by the people from the parasitology background, that these parasites, they sometimes they somehow play havoc with the populations, and a recent pandemic, the COVID-19, is one such example wherein the people had become, the humanity as such had become helpless facing this pandemic. That is how actually this parasite, if it goes out of control, if we are not in a position to somehow tackle these problems and somehow come up with certain suggestions and certain ideas and certain treatments, etc., then probably we may have to face tougher times. And since nature obviously is over there working at the background, somehow the nature has to have all of these entities in place so that a balance is maintained to run this universe. And it is the nature which actually takes the decisions, and the best decisions probably, which are important for continuing this race. But simultaneously, nature has given the humans a brain, and brain at the peak level, at the top level, so that the humans would think and then they would come up with solid ideas, so that if we are not in a position to eradicate the disease, at least we should be in a position, and we will be in a position to reduce the parasitic load, reduce the parasitic burden, and free the humanity of the ill effects of these parasites. That was the main idea behind having this integrated webinar on these two aspects, water on one side, parasite on another side, and in between we had health, because health finally remains the final target of the work which we scientists carry at our different levels. Without coming in between our learned speakers, which we have on the panel in today's valedictory, I would request Dr. Muslim to somehow carry the program forward. And I thank all the speakers, the panelists, the researchers who were part of this webinar and who helped us to make this webinar a success, particularly our 14 learned speakers who actually spent their valued time, and then the panelists on the inaugural function and the panelists of today's valedictory function. I am personally, and on behalf of the department and on behalf of the University of Kashmir, I am extremely grateful to all of them for sparing their valuable time. Wassalam, have a nice evening.
Thank you, Professor Fayaz, for your concluding remarks. And I will request Dr. J.K. Saxena, Secretary, Indian Society for Parasitology, to present his remarks on webinar. Dr. Saxena, please.
Dear Professor Chisti, Dr. Fayaz, Dr. Shabir, Dr. Arshid, Dr. Samiullah Bhatt, Dr. Shalja Singh, let me welcome all you to this webinar, who was conducted successfully in the University of Kashmir. Let me say a few things about the Indian Society for Parasitology. Although I missed the inauguration session, but today I asked Professor Fayaz that I will be saying something about the society. The Indian Society for Parasitology was established in 1973 with the purpose of advancing parasitology by providing a platform for interaction among scientists involved in different areas of parasitology. The society is affiliated with the World Federation of Parasitologists and is represented on the World Federation for Parasitologists Executive Committee as one of the 10 members. The Journal of Parasitic Diseases that contains the original article across the whole spectrum of parasitology is now being published by Springer from the year 2009. The society encourages the young scientists below 35 years of age by awarding Young Scientist Award for the best paper presentation during the annual conferences and Professor M.B. Mirza Award for the best publication of research work carried out in India. Besides these, there are 2 Oration Awards for eminent researchers and teachers. Dr. B.N. Singh Memorial Oration Award for the excellence in the research and Dr. B.P. Pandey Memorial Oration for contribution to teaching of parasitology. From this year, we are going to award a Professor K. Hanmantara Memorial Oration Award from 2021. It will be given to a scientist for his or her recognition or contribution in the field of biosystematics and diversity of parasites. The parasitic diseases continue to be a major concern to human and animal health in several parts of the world. Development of resistance in parasite and vectors to control measures genetic antigenic variations has affected the national and international efforts to control these diseases. Scientific meeting organized by the society provides a platform for the closer interaction between participants and advancing their academic outputs. These conferences organized by the society give ample opportunity, especially to young scientists, to exchange their views through individual and group discussion. I am hopeful that the deliberation during this international webinar on water, health and parasites have not only focused on the important developments in this field, but also bring out important recommendation to alleviate the human suffering to these problems. I actually would like to congratulate the organizing secretary for the successful organization of this webinar. Thank you.
Thank you, Dr. Saxena, for your valuable remarks. Controller of Examinations, Professor Irshad, will be joining us in a meanwhile, so I may move forward and like to invite our worthy Dean Academic Affairs, University of Kashmir, Professor Shabir Ahmed Bhatt for his presidential address. Professor Shabir Saab, please.
Am I audible?
Yes, sir.
My esteemed colleague, Dr. Fayaz Ahmed, Head Department of Environmental Sciences, my other colleague who is right now here, Dr. Irshad, Dr. J.K. Saxena, other distinguished experts, participants of this 3-day webinar on the most significant theme, water, health, and parasites. It's indeed a great privilege an honor for me to be a part of this valedictory function. I came to know about it last late evening that a 3 days international workshop is being organized by the Department of Environmental Sciences under the leadership of Dr. Fayaz Ahmed. First of all, I would like to congratulate him, for organizing such a webinar in which I see the experts from all over the world, across the globe, from United States of America to Japan to China to Malaysia, and then distinguished experts and participants from our own country. I was really delighted to go through the list of the participants and the deliberations and the topics that were covered and discussed during these 3 days webinar. So many congratulations to the director of the webinar and to the organizers. And in the capacity of the Dean Academic Affairs, I would like to express my deep sense of gratitude to all those experts who, despite their busy schedule, have managed to spend some time with our participants. I would like to begin with this way that this 3 days webinar on water health and parasites is one of the important, I would say, themes that he has chosen in 3 days webinar, because World Water Day basically became a United Nations Observance Day in 1993. And what I understand that it seeks to focus attention on the global water crisis. As a student of management, that's what my background is. Some of my colleagues are working in the field of management of water resources. And when we talk about water health and parasites, I think it is an area which concerns all of us, every individual across the globe, irrespective of the field that we come from. This is something, we all need to be concerned and worried about, and then contribute our bit in whatever capacity we can to address this issue. Ladies and gentlemen, as you know, that 22nd March every year, people, organizations, United Nations all across as World Water Day by taking actions primarily to take the water to tackle and to respond and to address the water crisis. And the idea back to 1992, the year in which the Rio Giro took place. These observances, basically what I understand every year, serve to reaffirm that water and sanitation measures are key to poverty reduction, economic growth, and environmental sustainability. Ladies and gentlemen, World Water Day is celebrated to raise awareness of 2.2 billion people living without access to safe water. Look at the African countries, look at many other countries, even some parts of our own country where we face and come across acute water shortage. And the number of the diseases that we every day read and come across, that are born, from the water crisis and the, other issues related to it. So what I understand that it's about taking action to tackle the global water crisis. And I think it's about time that we give a serious thought to this. And I think it's about time for all academic institutions and the universities all across the globe, that we sincerely make efforts, to and then forum groups and emphasize on scientists and experts in the field to really pay attention to this core area. Because it's a core focus of the World Water Day today is to support the achievement of Sustainable Development Goal 6, which is water and sanitation for all by 2030. So this day focuses its attention on the importance of fresh water and advocating for the sustainable management of water resources. This day, I think for all of us is an opportunity to learn more about water-related issues, which I'm sure over the 3 days webinar, by listening to the experts, must have at least, created an awareness about all these issues and be inspired to tell others, first of all, to tell our own self, to tell others and tell to the people that we come into contact with and take, some concrete action to make a difference in this particular building block of life. It's more than just essential to quench thirst or protect health. Water is vital, basically, for creating jobs and supporting economic, social, and human development. A new World Water Development Report, which is released every year on the World Water Day to provide decision makers with tools, with strategies, with approaches to formulate and implement sustainable water policies. This report is coordinated by UNESCO World Water Development Program on behalf of the United Nations Water. The annual theme for the World Water Day is aligned with the focus of this report. This year's theme on the World Water Day is about water, what water means to people, its true value, and how we can better protect this vital resource. The value of water is about much more than its price. The value of water has enormous and complex value for our households, culture, our health, education, economics, and the integrity of our natural environment. If we overlook any of these values, just give it a thought. All these values that I talked about, whether we are talking about culture, whether we are talking about health, whether we are talking about sanitation, the economics, sustainability, education, if we overlook any of these factors and the integrity of our natural environment, if we overlook, we risk mismanaging this finite and irreplaceable resource. Water is under, as you all know, when we go through the various reports, and the various journals and the studies that has been conducted in different scientific institutions, what I understand today is that water is under extreme threat from a growing population, increasing demands of agriculture and industry, and the worsening impact of climate change. As societies balance the demands of water resources, many people's interests are not being taken into account. That is something that we need to look at. Many water resources are under increasingly severe pressure from climate change and other global drivers. Climate change alters rainfall patterns, soil moisture, humidity, glaciers balance, and river flow, and all cause changes to underground water resources. At the same time, floods or droughts are rising in frequency and intensity over the next 40 years. It is reported that approximately 800,000 new residents will move to cities around the world every week. Look at the data, look at the, imagine the number, actually at the speed people are moving towards the cities. Population growth and rapid urbanization will create further pressures on water resources, and we will have a tremendous impact on the natural environment. Given these challenges, the need to manage fresh water properly, therefore, is very essential. And to study the different aspects, there's a huge responsibility on the part of the academia and the industry, to actually engage in a serious research so that we will be able to address to some of this issue. Sustainable water development is enshrined in 2030 Sustainable Development Agenda, with water-specific goals explicitly linked to other development targets. Ladies and gentlemen, fresh water is the most important resource for the mankind cross-cutting along social, economic, and environmental activities. It's the condition for all life on the planet and enabling or limiting factor for any social and technological development, a possible source of welfare, of misery, cooperation, or conflict. To achieve water security, we must protect vulnerable water systems. We must mitigate the impacts of water and relate to hazards such as floods and droughts, safeguard access to water functions and resources, and manage water resources in an integrated and equitable manner. UNESCO works to build the scientific base to help countries manage their water resources in a sustainable way through the intergovernment hydrological program around the world. But simultaneously, it also lays a huge responsibility on the part of all the nations to come up with concerted and concrete effort and the research proposed in the programs to see to it that how we can respond to these crises. Given water's role in mitigating and adapting to climate change, water could play a connecting role across the SDGs and across policy frameworks such as Paris Agreement. Ladies and gentlemen, let's come back to our own country, India has only 4% water resources, but approximately 16% of our population at the global, and therefore water protection and management in these is a big and a tremendous challenge. The efforts at the level of mass awareness, academic, research policies and planning, governance to be undertaken, therefore, are of paramount importance for addressing the issue of the water. Therefore, it's our duty to put our meaningful and actionable inputs before the government that should factor into political will. And that's something very, very important. That should primarily factor into the political will in the form of water-friendly legislation, policy, and planning. And that's where the responsibility is falls on the government and the legislation and the policymakers. Kashmir, primarily, as you all know, is considered to be a paradise on this earth. And as you all know, Kashmir is gifted with rich water resources in the form of lakes, wetlands, rivers, streams, springs, and glaciers. But the recent research suggested they are subject to degradation at various levels, which should be a cause of concern. And that's what we see when we look around what has happened to these water bodies over the last few years. I think it's a major concern for every Kashmiri, for all of us, and even at the state and at the national level. This international webinar, therefore, on water is therefore has come at the right time and a right platform to join the hands for academic and research collaborations on various issues of water, as it evident from the number of scientists from different parts of the world, like United States, Canada, Japan, Italy, China, Bangladesh, India, which are a part of this international webinar. Ladies and gentlemen, due to COVID-19 pandemic, the importance of water becomes much more important than ever size, because we have been listening every now and then the commercials that come on the television showing how to wash hands and how many times to wash and to keep personal hygiene to deal with the COVID-19 outbreak. So one of the important things here I want to kind of put across again, I'm reminded of what you call John Henry Newman, because that's very relevant to remind you about when he talks about the idea of a university, because all those people who are listening to me right now, coming from different academic, scientific, and institutions and the institutions of higher learning, especially universities, the professors, my colleagues from the University of Kashmir, I say it over and over again that there is a huge responsibility on the shoulders of my colleagues across the globe, to address to this issue, because university, primarily in the words of John Henry Mann, which he talked about in 1852, university is a place where students come from every quarter for every kind of a knowledge, a place for the communication and circulation of a thought, a place where inquiry is pushed forward, discovery is verified and perfected, errors exposed by the collision of mind with mind and knowledge with knowledge. And thus is created a pure and clear atmosphere of thought, which every student, every member of academia breathes. As an academic, I strongly believe that one of the important functions of the modern university, is to advance knowledge, engage itself in a hardcore research, in a cutting edge research. And I think, when it comes to the scientists at national and international level, and this is an area of water crisis, where we have to I would say, make investments, create infrastructure, I hope we are able to bring in the technology into an area, and my scientists, my colleagues, the university collaborate each other. I always say that no academic institution, no faculty, no university can exist in vacuum. We depend on each other to develop and progress. And the expertise which is available in different institutions of the world, in different scientific institutions, there is a need to collaborate. There is a need to enter into partnership. What Japan may have or what India has may not be available in China. What India has may not be available in Australia. So if we pool our resources, pool our minds, and I know there are brilliant minds and a lot of research, even in the University of Kashmir, I see my colleagues, even in environmental science, biotechnology, bioresources, and other colleagues, even some of my colleagues. I have a colleague, Professor Isaac, I would like to make a mention, who is actively involved in writing and looking at all these issues. Perhaps he's listening to me this time. And this is an area, because he's more into, I would say, into the pharmacy part of it, but I would ask my colleagues who are into it to come up with an engage yourself into an integrated research, and the kind of thoughts, the kind of scientific brilliant mind they possess. I think they can really collaborate into very nice, into very interesting research. Together they can work on the issues when it comes to the water crisis, because a lot of diseases primarily, whether it is jaundice or any other disease, they primarily emanate from water crisis. So I think this is relevant to medical, this is relevant to a biotechnologist, this is relevant to an expert of management who is interested to do some kind of research in management of water resources. It's relevant to economics as well. It's relevant to an industry. So this is an area where all of us can collaborate and put our minds together and come up certainly with some suggestions. I am sure that during these 3 days international webinar, looking at the kind of the experts with brilliant mind and the kind of expertise and the research they have conducted over a period of their time, it must have certainly, enlightened the participants, and they must have taken a lot of questions, a lot of lessons to home. And I hope that my colleagues at the University of Kashmir will continue to engage themselves in this kind of a research and organize these seminars, webinars. If nothing but at the end of the day, I always believe that it creates a curiosity, it raises certain questions, and we all go back, with some lessons to our home. It may not be as a scientist, but as an individual, it creates a sense of responsibility in my mind as to how I am supposed to use, how I am supposed to utilize the scarce resources, which is so important for me to look to see to it that, what would I do, in order to address to the major issue that's what we call, water crisis. So on this day, I think the best way, to celebrate and to mark this day would be to pledge. So let's all of us take a pledge on this World Water Day that we keep our hydrosphere safe for our sustaining living. And if we really take that pledge from the core of our heart, I think we will be able to make a difference, in our own individual and collective capacity. With these words, once again, thank you so much for giving me some time and for giving me an opportunity to be a part of this valedictory function on the most significant area, water health and parasites. I hope, as I said, I'm not a scientist. I don't come from hardcore science. I come from the field of management, which we believe is also a soft science, and this was my individual perspective, looking at it from the perspective of industry, economy, sanitation, health. And we have seen recently a lot of sanitation schemes have been launched by the government of India to create awareness among people. And I hope this seminar certainly would have created at least that awareness and contributed, and added to the existing literature and the knowledge, and the exchange of ideas among various scholars is always something, that takes this nation, that takes the field of academics, field of research forward. Thank you once again, Professor Fayaz Saab, organizers of this conference, making me a part and giving me an opportunity to speak my mind and to share some of my thoughts with all of you. Thank you so much. And I again thank you all for have been a part of this workshop, and to all the experts on behalf of the University of Kashmir in the capacity of Dean Academic Affairs, who actually spared some of their valuable time to be a part of this 3-day international webinar. Thank you so much. Thank you, Fayaz Saab. Over to you. Thanks a lot.
Thank you, Professor Shabir, for this exhaustive and wonderful overview touching the core theme of the webinar, that's water. Now I'd like to invite Controller of Examinations and Dean School of Biological Sciences, Professor Irshad Ahmed Nawji Saab, to share his words of wisdom to us and with our fellow participants. Professor Irshad, please.
Am I visible and hearable?
Yes, sir. You're audible. Your video is not coming.
Okay. Is it coming now?
Yes, sir, it's coming now.
Okay. At the outset, let me apologize that I was slightly late in joining the meeting because of some problems I faced on the way back to my home. And initially, I would like to thank all the members, distinguished participants, and the colleagues who have invited me for participating in this valedictory function. As the theme is, that is about the webinar on water, health, and the parasites. As majority of you may be aware that I have been working on a theme related to the important resources, that is the natural wealth of herbal medicine, medicinal plants. And obviously for that, whatever basic physical and biological entities play a role, they are dependent on water. And for that water sustenance, whatever other biological parameters we have to take care of. Unfortunately, we have seen that during the last few decades, the attitude, the proper caring mechanism, as well as the systems that are operative or that are in vogue are not at par with the requirements. I have been surveying almost all of J&K state along with other parts of Northwest Himalayas for looking at what the density and diversity of biological resources we have. During the last 2 decades, the scenario that has changed has sent a warning to all of us. What the initial stage was, what the initial greenery I have observed, what the natural resources I have observed in this particular belt, and what I see at present, there's a huge change. In the sense that what the density and diversity of natural resources was and what it is at present, it is not comparable. When we look at water, that is basically the backbone of almost all the living entities, and look at the status of different resources where this water was initially available, the changes that have occurred are depicting that perhaps the attitude of the dependent populations, especially people living in the vicinity of these habitats, have not been able to take care of the water resources. I have visited almost all the high altitude belts of J&K state as a whole, as well as other parts of Northwest Himalayas. The number of glaciers I have observed at the initial stages of my survey and the glaciers that I observe at present, there's a huge difference. On an average, what is estimated universally now, we lose roughly 20,000 glaciers per year. And the deforestation, in turn, is responsible for soil erosions. And whatever the other natural resource of water in terms of rivers, lakes, ponds, springs, etc. were, because of that attitude of destruction, the forest soil is getting eroded, and that is seeping into these water bodies. And because of that, the depth of rivers, depth of lakes, depth of ponds is decreasing. And for that, obviously, unless and until we assess the basic causes, basic reasons, I don't think anybody can conclude as to how we can be able to overcome such things. If we look at Dal Lake, one of the famous water bodies of Kashmir, or look at Wular Lake, one of the largest freshwater lakes of Asia as such, if we look at those and look at the areas that were measured about 20, 30 years ago, and the measurement that has been taken in around 2020, estimated area lost in Dal Lake is to be around 25 square kilometers. In terms of the encroachments, in terms of degradation of natural forest belts, in terms of soil erosion, in terms of seepage of that soil into these water bodies. And because of that, obviously, whatever diversity of living entities, be it plants, be it animals, be it microbes, was existent, that has in turn shown depletion. Similarly, if we look at Wular Lake or other lakes, the same is the scenario. Or if we visit the areas where initially we used to observe huge number of glaciers as sort of a natural wealth, and usually in the vicinity of those glaciers, whatever diversity of natural resources like plants, animals was seen to be present. If we compare the data of 20, 30 years ago with the present status in the vicinity of same glaciers or in the vicinity of same habitats, the number has decreased a lot. And that decrease in turn shows that perhaps it is not only a natural calamity, which we normally quote in almost every class, every lecture, that we are facing global warming. But if you look at the backbone of that global warming, primarily it is the dependent populations or dependent human communities who have played a negative role in terms of destruction of the dense forest lands or encroaching upon the belts where a natural wealth of biological resources was existent. And that encroachment has in turn caused degradation. And that degradation has resulted in the overall changes of the habitat features, and automatically the density and diversity of natural wealth of plants, animals, microbes has decreased. And because of that decrease, whatever other resources like water bodies, glaciers, etc. were existent about 30, 40 years ago, their number, their depth, and the quantity of water they had is decreasing. And obviously in that, the role played by us, that is the dependent entities, is reflective of the fact that our attitude perhaps has not been as good or as caring as it should have been. Otherwise, the number of glaciers, the number of water bodies, and the depth and purity of water in those water bodies could have been much better had we taken care of the resources as such. And there was a time when I was a kid, I was living in the vicinity of River Jhelum. My parents would never allow me to visit the river because the speed of water flow in that river, as well as the depth of that river, was so huge that they were afraid that I may perhaps sink in that. But at present, look at the same river, look at the same locations. You can see kids walking across the river without actually having any sort of fright that they may perhaps flow with water. Similarly, if we look at the lakes, look at Dal Lake, look at Uller Lake, look at Anchar Lake, and compare the purity of water, compare the depth of water, compare the quantity of water it contains, we find that almost all of that has got depleted. That shows that perhaps we have not taken care of water. And this water, in turn, is having a direct impact on whatever dependent entities use that water. It can be human populations. In majority of the cases, the people, communities living in the vicinity of these rivers, lakes, ponds, they sometimes try to consume the water. If the quality of water is not good, look at the impact of that water on the health of that dependent community. It can be human beings, it can be plants, it can be animals, it can be microbes who are using that contaminated water, and automatically their survival in turn shows that they are now getting starved because of the non-availability of pure water. Similarly, if the water is pure, whatever microbial wealth it initially had, which had a positive role, that would deplete or decrease because of the contamination of that water. And obviously, because of that, the parasitic wealth will positively increase because that may facilitate the survival of certain living entities. But that parasite, if consumed by a plant or invading a plant or a human body or an animal or insect body as such, automatically overall survival caliber or survival potential of that will get depleted. Now, even though the theme may have seemed to different members, what is the relationship of water health and parasites? If we compare the collaborative activities of those, we find that it shows the dependence. Purity of water is in turn reflecting the purity of health, and purity of the parasitic diversity will in turn have a positive impact. But if we have parasites of negative impact on the health, automatically whole biological community will in turn suffer a lot. And for this, obviously, it is not necessarily one should take out or carry out a work on his theme individually, unless and until people having a concept of assessing water, its purity, its contamination, and so on, or people looking at the health conditions of plants, animals, humans, insects, birds, or people working on parasitic diversity and their role, if they cooperate, if they collaborate and assess what would be a collaborative activity which will facilitate us not only to maintain and sustain the pure water, maintain and sustain the pure health, as well as try to help us to overcome whatever the parasitic problems we are facing, we cannot achieve the goals. So I hope during this webinar for the last 3 days, whatever discussions have taken place, whatever interactions have taken place through online mode, the suggestions communicated by different participants and the net outcome of those suggestions, once shared among all the participants, which in turn they can share with their own faculty or students or scholars, will help us to come to a conclusion where a collaborative activity of not necessarily a zoologist or a botanist, but a naturalist who have a concept of biology, who have a concept of nature as such, who have a concept of physical sciences also, if they combine together and derive a sort of a policy or a path with which they can in turn be able to draft a plan with which water, health and parasitic problems can be monitored and sustained at a level at which nobody in future will try to highlight as to what is the impact of impure water on his health, what is the impact of parasitic density and diversity on the survival of communities around that particular water body. We cannot be able to draft a plan, we cannot be able to implement that plan. Reason being that collaborative and cooperative activities help us to communicate our suggestions, our themes, our plans, with which whatever problems are arising because of, say, if I quote the same term, global warming, or because of the degradation of natural habitats, we may draft a plan which will help us to not only sustain what we have, but try to recover what we have lost during the last couple of decades. I hope the younger participants will carry with them a theme that unless and until we assess what are the causes of these problems that we are facing, that is problems about the water, purity, health problems or parasitic problems, we cannot be able to derive a policy or implement a policy to overcome such drawbacks so that our sort of future plans will help us to sustain it in a much, much better way and automatically help us to sustain whatever resources can help not only we people, that is the present communities, but the future communities also to use them properly, maintain and sustain them, and that will automatically try to revive whatever the overall natural biodiversity we had inherited, and that will automatically help us to draft plans so that future generations, not the present day scholars and students, but those who will try to join these themes of natural sciences will try to take care and try to in turn help automatically sustain and maintain whatever we are dependent on. I'm thankful to the organizers for inviting me to, say, participate in this valedictory function, but I will apologize. I could not actually participate in any of the sessions because of the heavy load. Majority of you are aware what sort of load I am at present handling. But I hope and pray that whatever little suggestions I have made, they will be shared properly with the participants and some sort of a conclusion would be drawn on the basis of whatever discussions, whatever deliberations took place during this 3-day duration, so that this outcome will help us to draft a policy which will not only maintain what we are dependent on, that is water, health, etc., but try to educate our progenies as to what plan they should try to follow in future, and that plan in turn can bear a fruit not only for us, that is present generation, but also for the progenies as such. I hope and pray that these few comments will be taken care of properly, and I would also at this time like to express my thanks, sincere thanks to Professor Shabir Saab for actually waiting for a slightly longer duration for my participation in this, as well as to my friend, my colleague, Professor Fayaz Saab, Muslim Saab, and Arshad Jahangir Saab, and other participants for allowing me to just give some concluding remarks about this workshop or webinar on the water, health, and parasites. I hope and pray that my comments will be taken slightly lightly, that in a short duration, I could not conclude in a dominant way, but hope and pray these few comments will try to help us to derive at a conclusion as to what should be the policy in future. Thank you very much.
Thank you, Professor Irshad, for giving us the insight on the state of natural resources, the theme of the webinar, and emphasizing the role of work for everybody to work. I may now invite Dr. Arshid Jahangir, Organizing Secretary and the Senior Assistant Professor, Department of Environmental Sciences, to present the vote of thanks. Dr. Arshid, please.
Thank you, Dr. Muslim. Bismillahir Rahmanir Rahim. Respected Professor Shabir Ahmed Butt, Dean Academic Affairs, University of Kashmir. Professor Irshad Ahmed Nauchu, Controller of Examination and Dean School of Biological Science, University of Kashmir. Professor J.K. Saxena, Secretary, Indian Society of Parasitology. Professor Fayaz Ahmed, Director, Webinar and Head, Department of Environmental Sciences. Dr. Samiullah Butt, Coordinator, Webinar. Dr. Muhammad Muslim, Organizing Secretary, Deans of various faculties, Heads and Directors of various departments and centers, esteemed professors, learned scientists, my colleagues, and dear participants. Assalamu alaikum wa rahmatullahi wa barakatuhu. It gives me an immense pleasure to extend the vote of thanks on the valedictory function of 3-day international webinar on water, health, and parasites, observed on the occasion of World Water Day 2021, which was organized by Department of Environmental Sciences, University of Kashmir, and Indian Society of Parasitology. A number of learned scientists of high repute from various prestigious institutions around the world have interacted with the participants and shared their knowledge, expertise, and recent scientific developments and advances related to various themes of the webinar. As proceedings of the international webinar are about to close, it is the duty of the host institution to extend the vote of thanks in favor of all those involved in making this webinar a real success. Quoting from our Prophet Muhammad sallallahu alaihi wasallam, "Man lam yashkurun nasa la yashkurullah." One who cannot thank people who have helped him a bit cannot thank Almighty Allah, who have showered unlimited blessings and favors to him. In this context and spirit, I, Dr. Arshid Jahangir, Organizing Secretary of the webinar, on behalf of the organizing committee, take the honor and opportunity to thank our Honorable Vice Chancellor, University of Kashmir, Professor Talat Ahmed, a scientist of repute, for presiding over the inaugural function. Professor Talat Ahmed has been always kind enough to accept our invitation and takes keen interest in organizing functions relevant to both academics and research. I also take the opportunity to thank Professor Sukhbir Kaur, President, Indian Society of Parasitology, for accepting our invitation to be the chief guest of the inaugural function. I am also grateful to Professor Shakil Ahmed Ramshu, Dean Research, University of Kashmir, and Professor Shamim Ahmed Shah, Dean School of Earth and Environmental Science, University of Kashmir, for their deep interest, continuous support, and valuable guidance for promoting this event. We are extremely grateful to Professor Shabir Ahmed Butt, Dean Academic Affairs, University of Kashmir, for readily accepting our invitation despite his busy schedule of work and grace this valedictory function with his presence and presidential address. Thank you, sir. We are also thankful to Professor Irshad Ahmed Nauchu, Controller of Examinations and Dean School of Biological Sciences, University of Kashmir, for being with us on this occasion. Sir, your gracious presence in today's valedictory function is a source of great encouragement to all of us. I am also thankful to Professor J.K. Saxena, Secretary, Indian Society of Parasitology, for gracing the valedictory function with his valuable remarks. I am highly grateful to all deans of the university, Registrar University of Kashmir, heads of various departments, faculty members, and other colleagues in the university for being with us during this webinar and providing logistic and facilitating in conducting proceedings of this webinar. I would like to put on record our special thanks to eminent guests, scientists, and other dignitaries who have participated from different parts of the world and India and have contributed in the webinar and made it fruitful and successful. With great enthusiasm, I thank all resource persons, namely Professor Zhang Fan, Institute of Tibetan Plateau Research, Chinese Academy of Science, Beijing, China, Professor Basant Maheshwari, Water, Environment and Sustainability, Western Sydney University, Australia, Dr. Amit Kumar from Nanjing University of Information Science and Technology, Nanjing, China, Professor Marco Cantonetti, Head Limnology and Phycology Section, Museo degli Scienze, Muse, Trento, Italy, Dr. Binaya Raj Shivakoti, Institute for Global Environmental Strategies, Kanagawa, Japan, Dr. Samindra Sherchan, Department of Global Environmental Health Science, Tulane University, New Orleans, USA, Dr. Larry Stevens, Director, Spring Stewardship Institute, Arizona, USA, Dr. Sara Naureen, Institute of Water Flood Management, Bangladesh University of Engineering and Technology, Bangladesh, Professor Mohyuddin Munawar, Fisheries and Oceans, President AEHMS, Burlington, Canada, Professor Ummi Shamim, Department of Zoology, Andhra University, Professor Kuldeep K. Sharma, Department of Zoology, Jammu University, Professor C.J. Hiwari, Department of Zoology, Dr. B.A. Marathwada University, Professor Shailja Singh, Special Center for Molecular Medicine, JNU, New Delhi. Let me express my gratitude to all participants who have joined from different parts of the world and showed keen interest and active participation and stayed with us for 3 consecutive days in this international webinar. I am quite hopeful that the deliberations may have immensely benefited them and given them exposure about the current scientific developments around the world in the field of water, health, and parasites. I express my special thanks to the advisory committee of the webinar, Professor A.R. Yusuf, Professor Neelam Gupta, Professor M.Z. Chisti, Professor Azra Nahid Kamali, Professor A.K. Pandit, Professor Kamal Jaiswal, Professor B.A. Ganai, and Professor G.A. Bhatt. We have been fortunate enough to be backed by a team of very motivated and dedicated teachers and colleagues and other staff members who know their job and are result-oriented, without which it was not possible to organize such a webinar of international standards. I am very grateful to the members of organizing committee who helped us in one way or the other, especially Professor Fayaz Ahmed, Head Department of Environmental Sciences, Dr. Samiullah Butts, Senior Assistant Professor, Department of Environmental Science, Dr. Muhammad Muslim, Assistant Professor, Department of Environmental Science, Dr. Asima Tanveer, Dr. Moiza Ashraf, Dr. Muhammad Irshad, and Dr. Shamsul Haq, lecturers in the Department of Environmental Sciences, research scholars and students who acted as rapporteurs, and other staff of the department for making this webinar a success. Thanks are also due to Director EMMRC, University of Kashmir, Dr. Salima Jan and her team for recording and disseminating the activities of the webinar. I gratefully acknowledge the PRO and his team for giving wide coverage to the webinar in both print and electronic media. Active support extended in diverse ways by IT department and other units of the university for providing all possible help and cooperation needed for the smooth running of the webinar is highly acknowledged. Special thanks goes to Mr. Tanmay Bardwaj, Account Executive, Zoom Video Communications, for providing the free Zoom platform for this webinar as a goodwill gesture. Above all, I am highly thankful to Almighty Allah who gave us enough courage to accomplish this task. Once again, I thank you all for being with us today. It's a great pleasure. Thank you very much.
Thank you, Dr. Arshid. I once again would like to thank you all for being a part of this valedictory session. Yeah, Dr. Arshid supposedly skipped Dr. Malhotra. I would like to, acknowledge his contributions. He was a worthy guest speaker. So I'd like to take an opportunity to once again thank you all for your participation throughout the webinar and in this valedictory session. I would like to announce that the participation certificate for the webinar will be mailed to the respective participants in the due course of time. Thank you very much. With these words, we'd like to conclude the webinar. Thank you. Have a nice day.